Okay, hi everybody, this is Lilith and I'm one of your IAs for BIPN 160, and this is my review video for lecture 9. So in lecture 9 we talked about taste and olfaction, so let's start with taste. So this is a tongue, and the tongue is the organ that has these cells that have receptors that respond to different taste stimuli, to different taste molecules. And so if we zoom in on the tongue, we will see that there are many different papillae, and each papillae has a taste bud in it. This is a taste bud, and each taste bud has 3 to 4 taste receptor cells. Okay, each of these cells will be releasing ATP as their neurotransmitter, and the ATP that they release will be stimulating the cranial nerve 7 or 9. Then this stimulus will be sent to the nucleus of the solitary tract. Then the pathway continues to the ventral posteromedial nucleus of the thalamus, and then the pathway continues all the way to the gustatory cortex. Okay. Now, how is this information transmitted? There are two theories: the theory of labeled line and the theory of population code. Labeled line is the theory that tells us that each type of taste cell, so each one of these guys, contains only one type of taste receptor, meaning there are only the same types of receptor on each cell, and that type of receptor responds to only one type of molecule. So in this case, the blue cell, for example, would respond only to sweet tasting molecules, and the red one would respond to, for example, sour tasting molecules. Okay, so that is what labeled line says. And I will talk a little more about that in a little. The population code theory states that each type of taste cell contains many types of taste receptors. And in this case, if we zoom in over here, this theory states that this one cell contains many different type of receptors. So this red one over here might be responding to sour taste, and this blue one might be responding to sweet taste. Okay, so those are the two theories. Now, the one theory that has been supported and confirmed over and over is the labeled theory, labeled line theory. And how has this been supported? Well, many experiments have been done to support the labeled line theory. The first one is that researchers took the different taste receptors and they knocked them out, meaning they disabled them or made them non-functional. Okay, and after they made each taste receptor non-functional one at a time, they were able to record the neuronal activity that is coming from the neurons that are receiving information from that specific receptor. And the results were this: if they knocked out, I'm not sure which one this is, umami. So this one is the receptor for the taste umami. So if you knock this out, meaning you make this receptor non-functional, and you take neuronal activity from all of the neurons in the pathway, you can tell that it is only the umami neuron that no longer has neuronal activity. This confirms the fact that there is labeled line in the gustatory pathway, because no other taste pathway or no other neuron that responds to other taste receptors was affected, and only the umami was affected. Okay, and there's another, for example, in this case, for the sweet receptor, if the sweet receptor is knocked out, then only the neuron that carries that sweet sensation no longer has any neuronal activity, and every other one is still active. Okay, so I've summarized that for you here. It confirms that each receptor sends sensory information to specific areas in the cortex via a specific neuron. Okay, you can think of labeled line as driving down a road, let's say taking the road from here and going all the way to Los Angeles. Imagine there were no emerging traffic, or there was no way for you to drive off of that highway, right? The only way you could get from here to there was to get on the highway and just drive straight. So that is labeled line theory. Okay. The next experiment that was done is depicted over here. And here what they did is they took a receptor that usually does not come up in the mouse tongue. So the mice usually don't have this receptor. And they expressed this receptor. I think it is the T1R2, you don't need to know, but just know that they expressed the receptor in the tongue of the mouse. And when they gave the mouse a water that had a concentration of this phenyl compound, which is the molecule that activates that receptor, then certain receptors were more active than others. For example, when they expressed this receptor in a sweet type of neuron, so a neuron that usually contains a lot of sweet receptors, then the mouse would start to drink a lot more of this water because that sweet receptor was activated, and the mouse likes to drink sweet water because it thinks that the molecule that is activating the sweet receptor is a sweet molecule. But in reality, it's not a sweet molecule. It's just a regular non-tasteful molecule that is activating the sweet receptor. Okay. However, if that same receptor was expressed in the cells that contain bitter receptors, the mouse would stop drinking the water because the mouse would think that that molecule tastes bitter. And again, in reality, the molecule doesn't taste bitter. It is only activating the bitter receptors. So the mouse's brain thinks that whatever that molecule is tastes bitter. Okay, and so this is another research study that showed that whatever receptor that you activate in the tongue, that sensory information is being sent to a specific part in the cortex that tells us exactly what type of molecule is activating which receptor. Okay, and that again is consistent with the labeled line theory. There were a couple of imaging studies that they did. For example, in this study, I pulled it from the lecture notes, they used calcium imaging in the nucleus of the solitary tract to show that indeed in the solitary tract there are different distinct areas where different types of tastes are being sensed. Okay. This image just shows us beautiful colors, and it shows the cortex by fMRI. And I called this, you probably shouldn't call this, but I just call it, quote, gustatory map, because this tells us exactly which area of the cortex responds to which type of taste. And again, this confirms labeled line theory because we know that we have receptors that respond to bitter, salty, sour, umami, sweet, and I guess carbonation also. And we see here exactly by using fMRI, we see in the cortex that each sense or each taste goes to a specific part in the brain. Okay, and then the last research study that was done to confirm the label line theory of the gustatory pathway was not to inhibit the receptors or simply watch by and some type of imaging. Instead, what they did is they stimulated different areas of the cortex and watched the behavior of the mouse. So if you remember again, this concept of required versus efficient or able to, sufficient or able to, is very important for this class, and you should study it for this exam number 2 also, because you will need to use different techniques to figure out if an area of a brain is required or sufficient. So to reiterate, if you're trying to figure out if an area of a brain is required for a certain function, you want to somehow either lesion that area or inhibit that area using optogenetics or chemogenetics. And if you inhibit that and the function stops, then you can say, yes, this area of the brain is required for this specific function. If you inhibit the area and the function keeps on going, so the function does not change, then you can safely say that this area is not required for the function. Okay, so that's the required experimental thinking that you can use. If you want to see if an area of a brain is sufficient to carry out the function or is able to carry out the function, then you want to stimulate that area and see if the function indeed becomes stronger or there's more of that function. Okay, and if there is, then you say, yes, this area of the brain is sufficient enough or is able to perform this function. So in this case, what the researchers did is they stimulated different areas of this gustatory cortex, okay, the taste cortex, and by doing so, I forgot what they did. Sweet cortex. Sorry. They stimulated the sweet cortex and they stimulated it in these areas, or I guess just in this box, in this chamber 1. And because it was stimulated, the mouse hung around in this chamber more because it just likes the sweet taste. And then when they stimulated the bitter cortex, the bitter area of the taste cortex in chamber 1, you see how the mouse no longer was staying in this chamber and it started moving and hanging around more in here. So that means that the mouse didn't like the bitter taste that it was being stimulated for. Okay, and so this study yet again confirmed that there are different areas of the brain that respond to different tastes, for example, sweet and bitter. Okay, now let's move on to olfaction. So olfaction is the sense of smell, and so we can smell different types of smells through the I forgot what this was called. This, I think, was called retronasal, and this was called... sorry, you have to look at the slides because it has escaped my mind. But there are 2 pathways to get to the sensory cells, basically, through your nose and through your mouth also. Okay. I'm going to zoom in over here because at the top of our nose we have this bone that is called the ethmoid bone. Okay, and in the ethmoid bone there's this area that is called the cribriform plate, and there are many tiny holes in this bone, and these holes are openings for the olfactory sensory neurons to be hanging down into our nasal cavity from the olfactory bulb. Okay, now let's zoom in over here. I took this image from the lecture notes, but then I flipped it so it's easier for you to see that this is the orientation of the cells. So this is the cell. At the bottom here we have cilia. And then these cells go up into the olfactory bulb, which sits right here. And in the olfactory bulb, we have glomeruli. And we will see this glomeruli in a couple minutes. Okay, these cilia are these little cilia right here, just more zoomed in. And these cilia contain receptors on them. Okay, and these receptors will be responding to different types of molecules. This is a more colorful image, so I flipped this one also so that you can see the cilia over here at the olfactory epithelium, and also remember that the olfactory bulb is right here. Okay, so each olfactory neuron will have the same types of receptors, just like the gustatory system. Okay, so at the periphery, so at the peripheral part of this pathway, at the nose, this seems like it is a labeled line system. Okay, again, why does it seem like it's a labeled line system? Because every olfactory neuron contains the same types of receptors. For example, these red receptors are only on the red olfactory neurons, okay, and so on. However, the olfactory system has something that is a little more unusual. The odorants, the molecules that we smell throughout our day, they have, obviously this is a molecule, right? So one part of the molecule, this red part, will be recognized by a receptor, and then this green part of the molecule will be recognized by another receptor. So the red one will be recognized by this receptor, the green one by this receptor. And by doing so, the olfactory system is called a combinatorial system, because different receptors can respond to the same odorant. And that will give us like a well-rounded idea of what exactly we are smelling. Okay, then let's follow the arrows that I have here. After picking up the sensory information, so after picking up the sensory information through the cilia, the neurons that look similar to each other, meaning that they have the same receptors on them, they converge into their own glomeruli. Okay, glomeruli is plural for glomerulus. Now, glomeruli lie right at the top of the cribriform plate, inside the olfactory bulb. And the glomerulus is just a bulb of synapses. Okay, so if I zoom in over here, you can see that each type, so let's only look at the red ones. Each type of olfactory neuron, right, all these red ones contain the same type of receptor on them. And because they have that same receptor, they all converge to the same glomerulus, and they all stimulate the same neuron that goes into the brain. Okay, so far we're seeing labeled line theory. Okay, no population code just yet. Okay, after each glomerulus provides synapses to individual neurons in the olfactory tract, so after each glomerulus gives these synapses to one neuron in the olfactory tract, these tracts go to the brain. Okay, so far again labeled line theory. And this is just, I think, calcium imaging showing how in the olfactory bulb there are different glomeruli that respond to, they're kind of bunched up, right? So there's some type of organization, okay? Still confirming labeled line theory. But then there was an experiment that looked into the structures that are much deeper into the brain. Okay, so this researcher and their team said, well, that's cool. We got to the olfactory bulb and it's still labeled line theory. But what happens when the olfactory bulb actually projects to deeper areas of the brain? And when they looked into this, they saw that when the olfactory bulb projects to the piriform cortex, there is actually no organization to these projections. Okay. However, when the olfactory bulb projects to the cortical amygdala, there is like a cluster of where these neurons end up. Okay, so they ended up just saying that the piriform cortex is disorganized in its input that it is getting, whereas the cortical amygdala is very organized. Okay, and so from this rose the idea that the piriform cortex, it can actually be population code, and the cortical amygdala is labeled line theory. Okay, so that's just here. Input to cortical amygdala is organized, so it is labeled line, and input to the piriform cortex is disorganized, so it is population code. Okay, and then the researchers asked a question of, can the piriform cortex be involved in associational learning? Okay, and I didn't include all of the images from the researches, but I summarized it in a way where it should give you the main ideas, but you should still look at the images if you want to a deeper understanding of these research studies. But basically, what the key points that I think you should know is that they thought that the piriform cortex might be involved in associational learning. And to test that, they inhibited the cortex, the piriform cortex, and they tried to see if the learned behavior is gone, and indeed it was. When they did inhibit the piriform cortex, then the mouse no longer remembered what it had learned by associating odors with a behavior. Okay, so that's on the piriform cortex. On the cortical amygdala, they asked the question of maybe it is involved in hardwired or innate behaviors. And the word innate, for people who might not know, innate means something that is from our nature. So if like I see a tiger, I'm innately going to be scared and I would want to run away. Okay, and same for the mouse. If the mouse sees something scary, it will be afraid and not want to go to that area. I lost my train of thought, but basically piriform cortex thought to be involved in associational learning, and cortical amygdala be involved in innate behaviors. And the way that they could test whether the cortical amygdala is involved in innate behaviors or not, they expressed halorhodopsin in the cortical amygdala. And halorhodopsin, as you know, silences this area, so it shuts up this area. And after they did that, the mouse was no longer avoiding the corner of the chamber that smelled like TMT. And I think TMT smells like the urine of a cat or something, or maybe I'm thinking of a different class. I'm not sure, but TMT is a molecule that scares away the mouse. So when they expressed halorhodopsin and they shut off the cortical amygdala, they realized that manipulation to this area of the brain made the mouse completely not even care about the smell of TMT. Okay, So the mouse was no longer innately afraid. So this type of experiment, hopefully you're already catching on to the required / sufficient experiments. This kind of experiment told the researchers that the cortical amygdala is indeed required for the mouse to be innately afraid of TMT. Okay. Then the researchers wanted to do the other type of research study, right? The sufficient or able to one. So they expressed and activated channel rhodopsin in the cortical amygdala. And this showed that the mouse actively avoided a certain corner of the chamber. Again, because that was the result, you can safely assume that cortical amygdala is sufficient or is able to initiate innate responses. Okay. The last study that Dr. Ru talked about, I think this was the one that he did in his lab. I didn't include too many details, partly because I'm tired and partly because he kind of also jumped through it. But I would definitely say, if there's one thing that you're going to remember from this study or from this talk, know that the relay to the nucleus accumbens drives attraction, and that the relay to the medial amygdala drives aversion. Okay? I hate doing this, but this is something that you want to memorize. Okay? And if you have any questions, we can definitely go over this. But I would say for the exam, to succeed, if you're given a question like this, memorize these 2 bullet points. Okay? I kind of flew over that because I also have to study for exams, but hopefully that was a little more help, a little more explanation on these topics. These I posted here, these questions, and the answers are already here, so I won't go over them, but please go over them. This is good practice. And also this one, you can stop the video and read it, or you can also find this PDF in my IA page. And I've also included the learning objectives for you here. Okay. That's it for this video. That's it from me until after exam 2. I will be kind of busy tomorrow. I have 3 exams and then they're all back to back before your exam. But if you really need to ask me a question, shoot me an email and I'll do my best to reply it, but I might not guarantee it. Okay, but regardless, if you're on the Discord channel, ask away. Some of the IAs will be available to answer your questions. And one more thing for the exam, don't be afraid to ask a question on the discussion board or to email us or the professor if you want to do it privately. But if there's a question that you're not understanding or anything, please ask, because you don't want you just not understanding the wording to be the problem, the reason why you don't get it. Okay, so don't be afraid to ask. All right, that's it from me. Good luck, everybody, and I'll see you after.